Hello, everyone. Welcome to the interviews of the South American Mycorrhizal Research Network. My name is Camille Truong, and today I have the pleasure to be with Jonathan Plett, who is a professor at Western Sydney University. Welcome, Jonathan.
Thank you.
Thank you for being with us today. And so we are going to talk about a review article that you recently published in the journal ISME, entitled Leveraging Genomics to Understand the Broader Role of Fungal Small Secreted Proteins in Niche Colonization and Nutrition. So first, can you tell us a little bit about what are these small secreted proteins and why you were interested to study them?
So to me, small secreted proteins are the language of fungi. So fungi, like us, communicate with other organisms in their environment, and they don't have vocal cords, right? And so they use a range of different things. So they use chemicals that are called metabolites. They use these proteins that are quite small. And other things like that. And so I'm interested in these proteins because the language, I would say, that they are is a way that they are able to broadcast that they are there, but more interestingly, they affect how other bacteria, other fungi, other plants go about their normal life.
And so there have been quite a few studies looking at how these small secreted proteins influence host in parasitic, but also mutualistic fungi. So what are the mechanisms involved? Can you tell us a little bit about this?
Yeah, so we know that these small proteins are able to interfere interfere is probably a strong word, but are able to influence a lot of different processes in plants. And so the way they do that is by binding to other proteins in the plant and completely either turning them off. So proteins in humans and in plants and in fungi have all range of different roles, involved in metabolism and finding where nutrients are and things like that. And so what these fungal proteins that I studied do seem to be able to influence the immune system of a plant, and to either turn it off, because the fungi I deal with are beneficial. And so these proteins go out into the plant cell or stay on the outside of the plant cell, and they turn off that immunity in the plant to allow that fungus to colonize. And then once that fungus is there, it actually gives nutrients to the plants and it helps it grow.
So do you mean that the fungus, the mutualistic fungus, is manipulating the plants?
Totally.
Is that what you mean?
Yes, it totally manipulates.
And so how do you think the plant respond to this? Because this is a mutualistic interaction, right? So we often talk about the benefits for both. So in this sense, do you see it more as a parasitism kind of interaction, or what is your point of view about this?
Yeah, it's a good question. It depends honestly on the day that you ask me. It almost like, it sits on the fence, right? It does have benefit for the plant, and so we know that. So it can't be really classified as a parasite, but it's using some of the same machinery that a disease or a parasite would actually use to manipulate that plant host. So it's almost like it's evolved or learned some of these similar strategies as a disease-causing fungus does to manipulate a plant in order to colonize and at least get its foot in the door, right? It's almost like it knocks at the plant and the plant is like, what? And the plant kind of opens the door a little bit to allow the fungus in, and the fungus uses these secreted proteins to be able to stop the plant from immediately rejecting it until some of that nutrient can flow.
Okay. And in your article, there are several research that have been done showing that these small secreted protein, they seem to be present not only in the genomes of parasitic or mutualistic fungi, right? But they are found in like saprophytic or early lineage fungi. And so how would you explain this if they don't really interact with a plant host?
Yeah, that's something that's really only been evolving in the last few years, or sorry, not evolving, but our understanding of that has really only been coming out in the last few years, because initially these proteins were only really thought to be in disease-causing fungi, and then they were found in these mutualists, they were found in bacteria, etc. But now that they're being found to also be in saprotrophs, it does really lead to that very difficult question of, okay, we spent years, if not decades now, saying these manipulate the plant host. But when you find them in a fungus that doesn't manipulate a plant host, as far as we know, that kind of makes you scratch your head. And I think, to me, what that says is, and what we say in the article, is that these fungi or these protein sequences have evolved over time. So probably they had some base role that wasn't in communicating with a plant, that might have been, accessing a substrate or accessing another nutrient, or maybe even influencing bacteria in the soil that we don't know where to look yet. And that over time, random mutations have led to the ability of these proteins to interact with the plant host. And that's probably what gave birth to the mutualistic lifestyle in some of these lineages.
Okay, but so do you think they could have more general role for the nutrition of this fungi in particular?
Yeah, so it's been shown in other labs that actually some of these proteins are involved in communicating within the fungus itself. So it secreted from one hyphae and then taken up by another one. So it can actually act as communication within the fungal network itself. Other research is showing as well, and our work is starting to show the same thing, that it looks like fungal proteins might be interacting with bacteria in the soil and changing how they actually act as well. So it could be that saprotrophic fungi are also using small secreted proteins, not to manipulate a host, plant host, but actually to manipulate other microbes in the soil.
And so, of course, because there's small secreted protein, they're secreted in the environment itself is going to have a large influence on the activity of these. So have you done some studies about this? What can you tell us about this?
So we haven't done studies yet on how abiotic environments are influencing things, other than we have looked at when we are altering the fertilization within the soil. And in that case, the mutualistic fungi actually change very dramatically how they are acting. And some of these proteins are actually turned off or turned down very low. We've also shown in our work that different isolates of the same species have different complements of these proteins. And when we look in the microbiome of the soil around these hyphae, we find that different isolates have different microbiomes themselves. So that's leading to us asking the question, and I don't have an answer yet, but are they actually, these mycorrhizal fungi, influencing their microbiome around them using these small secreted proteins as well?
And do you think that this variability that you find in this small secreted protein could also influence I don't like to talk about host specificity, but maybe the host preference that some fungi would have for certain plants?
Yeah, very definitely. Yeah, so we have a project ongoing at the moment looking at just that question. Because again, leading out of some great work from Sophie and Camoon's lab, they were able to show that with Phytophthora, even one amino acid change within an effector or one of these small secreted proteins, that was able to increase the host range of that disease. And so really we're coming down to really fine nitty-gritty details of being like, oh, it might not be just protein presence absence, but it's actually protein sequence that might influence host range of these mutualistic fungi.
Excellent. And so if someone who works with fungal genomics would like to study this, what would you recommend for them to do? Are they difficult to detect first in the genome?
They're not difficult to detect in the genome. That part is thankfully one of the more simple parts. I mean, they all have a secretion domain that we've been focusing on mostly, though there are non-classical secreted proteins. depending on the genome, there is a huge range. So the ectomycorrhizal fungus Leucaria bicolor probably has between 1 and 200. The ectomycorrhizal fungus Physolethus that I mostly work with now probably only has between 10 and 20. So, I mean, you do have large variations in what we would call host-induced small secreted proteins. They do have more secreted proteins than that. And so the ones we would think would be involved in communication, that's kind of the number range that you're looking at. So genomically, you can look at that. Comparative genomics are phenomenal at the moment in the fact that we're beginning to trace the lineage of those. And I think that's one of the next steps that we have to focus on is where are they actually found within the fungal tree of life? When have they evolved? How have they evolved? And then it's just getting into the lab and start really characterizing them. And that's where the difficulty comes in and where the time and huge person effort comes in.
Yeah, I imagine. So finally, can you tell us a little bit about your own research on this that you're doing at the moment?
Yeah, so our group is looking at a number of things. So one of them actually is what you mentioned earlier is how dependent on the secreted proteins is the host range of an ectomycorrhizal fungus. So we're looking at Physolethus with that, and that's an Australian Research Council funded project. And we're also looking at the reverse side now too. We're also interested in looking at, well, if the fungus is manipulating the plant, obviously there must be, well, maybe not obviously, but maybe there's the reverse. And so we had some initial work published a few years ago in scientific reports that would show that populus roots are able to secrete small proteins as well that seem to influence the fungus. So we're looking at that in eucalyptus as well now. So those are the main drivers at the moment, is really understanding what secreted proteins are there, what are they doing, and how essential is their sequence to host specificity.
Okay, thank you very much for your time. And we will be back soon with more interviews for the South American Mycorrhizal Research Network.
Thank you very much.